Welcome back to the deep dive. We spend so much time talking about the amazing ability of the human body to fight off external threats. Viruses, bacteria, all the stuff that makes us sick.
The soldiers, right. The
Yeah.
assassins, the frontline defense. The immune system is justly famous for its capacity to kill invaders.
But today we are talking about a far more sophisticated problem, a really high-stakes security crisis. What happens when the system turns in on itself? I want you to just step into a scenario that is, well, it's every parent's worst nightmare.
Okay.
Imagine holding an infant, maybe just 6 months old, and watching their body slowly, catastrophically just dismantle itself.
And this isn't science fiction. We're talking about a severe, totally unexplainable failure of the body's most basic security programming. The baby suffers from what doctors call autoimmune polyendocrinopathy.
Which means,
It means their immune system is attacking multiple vital systems all at once. The gut, which leads to massive digestive failure. The endocrine glands, causing a total hormonal collapse. The skin, resulting in severe, just unmanageable inflammation.
the official name for this is IPEX syndrome, immunodysregulation, polyendocrinopathy, enteropathy, X-linked syndrome. And historically, it was a death sentence.
A death sentence, yeah. Total systemic failure within weeks or maybe months, just because doctors had no way to stop this rogue army from destroying his own castle.
And this wasn't just some super rare thing only seen in human cases. For decades, researchers had an almost identical mystery playing out in their labs.
In a strain of mice.
The infamous Scurfy mouse.
Yes, the Scurfy mouse.
Yeah.
It was named that because its skin got all scaly and terrible from this uncontrolled, massive multi-organ inflammation and autoimmunity.
Sounds kind of cartoonish, but it was deadly serious.
Oh, absolutely. The disease was swift and lethal. It killed the mice just as quickly as IPEX kills infants. And for decades, the cause of this self-destruction, the identity of this failure, was a complete unknown. The soldiers had gone rogue, and nobody knew who was supposed to be in charge of stopping them.
It's the ultimate paradox, isn't it? The most powerful fighting force on the planet, your own immune system, failing at its most fundamental job. Telling self from non-self.
What is me and what is the enemy?
Exactly. And when that distinction collapses, the result is just tragedy.
But the mystery, as you said, is finally solved. And the world really recognized how profound that solution was. The 2025 Nobel Prize in Physiology or Medicine was awarded to three incredible scientists, Mary Brunkow, Fred Ramsdell, and Sakaguchi, for cracking this exact case.
And they didn't just find like a random gene or some obscure mechanism. They identified the actual cellular peacekeepers, the fundamental security guards that prevent all of us from developing these catastrophic autoimmune diseases every single day.
These are the regulatory T cells, or as everyone calls them, Tregs. They are the secret to what we call immune tolerance.
So today, we are diving deep into that quest. How did this trio of scientists solve the paradox of self? And what exactly is the molecular blueprint they defined for what's called peripheral immune tolerance? Our mission is to understand these peacekeepers and their extraordinary playbook.
To really appreciate the genius of what Sakaguchi, Brunkow, and Ramsdell did, you have to remember what immunology looked like before their work. Specifically, how the scientific community thought the immune system learned restraint in the first place.
Right, so before them, everyone was focused on this primary safety net, what they called central tolerance.
Think of it like this. T cells are born in the bone marrow, right?
Mm-hmm.
But they go to boot camp in a little organ called the thymus. Central tolerance is the first line of defense. It happens right there in boot camp.
Okay, so they're in training.
Exactly. And during that training, if a young T cell shows any sign, any hint at all, that its main weapon, its T cell receptor, recognizes or wants to attack self-antigens.
Meaning any part of its own body.
Yes, any part of me. If it does that, it is ruthlessly eliminated. Programmed to die. Apoptosis. Gone.
So we've used this analogy before, and I think it's perfect here. The thymus is like the sports draft.
The NFL draft, yeah. I like
The
that.
coaches are paranoid, right? And if a player, even accidentally, tackles their own teammate during a scrimmage,
They're cut immediately.
instantly, they are cut from the roster, eliminated on the spot. That's central tolerance. It's all about preventing the bad players from ever making the team in the first place.
Exactly. And for decades, the prevailing scientific mindset, and this is so key, was that this central process was the only thing preventing autoimmune catastrophe. If you developed an autoimmune disease, it must mean the thymus elimination process had failed. spectacularly.
Which, just feels like an insufficient explanation. It doesn't really account for how common autoimmune diseases are. We know bad apples slip through the draft all the time. Our bodies are just too complex.
They absolutely do slip through. And that's where Shimon Sakaguchi steps onto the scene in the mid-1990s. He's basically telling the entire established scientific community that they're missing half the picture.
He was really swimming against the tide.
Completely. He was proposing the existence of a second, crucial layer of control, a failsafe that operates out in the field, away from the Thymus Boot Camp.
A second line of defense. So not just cutting players at the draft, but having police on the field during the game.
The internal police force. That's a perfect way to put it. Peripheral tolerance.
And this implies the immune system isn't just passively trained once. It has to be actively managed all the time out in the tissues.
Yes. And that was a radical idea, because it meant the immune system wasn't just about eliminating danger. It was equally about actively maintaining peace.
So why was everyone so resistant to this idea? It seems logical.
Well, for one, it's metabolically expensive. To actively maintain peace, you need a whole separate army just to suppress your own soldiers. It just seemed inefficient.
And they hadn't found the cells yet.
Right. The thinking was, if these Peacekeeper cells exist, why haven't we seen them? Where's the evidence?
But the clinical reality, what was happening in hospitals, it just spoke louder than theoretical immunology. The cost of this safety net failing is just, it's devastating. It's chronic and destructive.
Absolutely. I mean, think about the diseases we see when this control is lost. Type 1 diabetes, where T cells destroy the insulin-producing beta cells in the pancreas.
Gone.
Or systemic lupus, SLE, where the body creates these autoantibodies that attack multiple organs, kidneys, joints, skin. Or rheumatoid arthritis, the painful destruction of joints.
And things like inflammatory bowel diseases, like Crohn's and ulcerative colitis.
The common thread in all of these is an army gone rogue, an uncontrolled system. And before these discoveries, the tools doctors had to deal with it were frankly terrible.
It was a scorched earth strategy. That's what the sources called it.
A perfect description. If your body was attacking your joints or your pancreas, the treatments involved broad, non-specific immunosuppression. We're talking high-dose steroids, powerful cytotoxic drugs.
You're essentially nuking the entire immune system from orbit just to stop one rogue battalion.
You are. You're damaging all of your defense mechanisms globally just to stop a highly localized attack. And that, of course, exposes the patient to a severe risk of infection, cancer, and just significant long-term complications.
It's like having to turn down the volume on the entire stereo just because you can't find the specific knob for the one instrument that's too loud.
Yes, you have to mute everything. But Sakaguchi believed there was a specific knob. He believed he was held by this invisible police force, and that's what launched the quest to find them.
So Sakaguchi knew this peripheral peacekeeping force had to exist. But, how do you find one specialized type of cell among the trillions of T cells already circulating in the body?
You need the uniform.
Right. You need some kind of physical signature, something that makes them stand out.
Exactly. So this brings us to phase 1. The initial marker, back in 1995. Sakaguchi's team performed this really key experiment. They began sorting T cells, just looking for anything that was different. And they noticed a distinct subset of the helper T cells, the CD4 plus T cells, that consistently expressed extremely high levels of a specific receptor subunit.
And this was CD25.
CD25. Now, this is the alpha chain of the receptor for something called interleukin-2, which is basically T cell growth fuel. We'll get to why that's so important later. But for now, just think of it as the visible patch on the security guard's uniform.
Okay, so they found the patch.
They found the patch. So Sakaguchi did the definitive experiment. He took mice, and he selectively removed only the T cells that were wearing this CD25 patch.
And what happened?
Catastrophe. The mice immediately developed severe fulminant autoimmunity. They looked just like the Scurfy mice and the IPEX infants.
Wow.
Then they did the reverse experiment. They took those CD25 positive cells from a healthy mouse, and they injected them back into the sick autoimmune mouse.
And the disease just vanished.
It vanished. They restored tolerance. Order was restored.
That is about as definitive as proof gets, isn't it? The cells wearing the CD25 patch were the security guards. You take them away, the peace collapses. You put them back in, order is restored.
It was groundbreaking. It was the first solid evidence for the existence of these suppressive T cells, but it still didn't explain why they were suppressive.
Right. Was CD25 the cause, or is it just a location tag? Like, was it just the uniform, or was it also the weapon?
Exactly. It was a marker, but it wasn't the instruction manual.
Which brings us to this parallel, totally unrelated detective story. Phase 2, The Genetic Culprit, which was in 2001. This was led by Mary Brunkow and Fred Ramsdell. They weren't even looking for CD25. They were looking for the single genetic flaw that doomed the Scurfy mouse.
And you have to remember, this was a painstaking effort. This was the late 90s, early 2000s. We didn't have the rapid, cheap, whole genome sequencing we have today.
Oh man, no. Mapping a single mutation was a colossal undertaking.
It was. They knew the Scurfy phenotype, that uncontrolled lethal inflammation, was passed down through a mutation on the X chromosome. So they were literally mapping and sequencing individual fragments of DNA, piece by piece, just trying to find this tiny flaw in the blueprint.
A needle in a genomic haystack.
Pretty much. And after years of dedicated work, they pinpointed it. The single culprit gene responsible for the catastrophic immune failure, a gene called FoxP3, forkhead FoxP3. When this one gene was mutated, the immune system lost all control.
And here's where the two detective stories just slam into each other. What did they realize when they looked at humans with that same problem?
They found that mutations in the human equivalent of the FoxP3 gene caused that fatal rare pediatric syndrome we started with.
IPEX.
IPEX.
Seriously?
Yeah.
The exact same genetic screw-up that killed the mouse was the same one causing this specific catastrophic human autoimmune disease. That's a shocking convergence.
It was. It established FoxP3 definitively as the molecular switch for immune tolerance. Without a functioning FoxP3 gene, you have no immune control. The defense system just self-destructs.
So let's just recap. On one side, you have Sakaguchi, who identified the cells by their uniform by CD25. On the other, you have Brunkow and Ramsdell, who identified the master genetic instruction manual, FoxP3, by tracking a legal disease from mice to humans.
Which culminates in Phase 3, the definitive link, in 2003. The science converged. Multiple labs, including Ramsdell, Sakaguchi's, and another scientist named Rudensky, all confirmed the same thing at once.
What was it?
The suppressive CD4 plus CD25 plus cells that Sakaguchi found always express FoxP3. FoxP3 was the code that commanded the cell to put on the uniform.
So the final official classification of these regulatory T cells, the Tregs, was established. They are CD4 plus CD25 plus FOXP3 plus cells.
FoxP3 is what we call the lineage determinant. It's the master regulator that decides the T cell's identity and its function. It literally programs it to be a peacekeeper instead of soldier.
And this discovery gave us, for the very first time, a molecular handle on peripheral immune tolerance, that fundamental system that keeps you from attacking yourself long after you leave the Thymus Boot Camp.
That shift is just monumental. We're no longer stuck asking, why did the boot camp fail? Now the question is, how do we support and control the specific security guards who are responsible for maintaining order out in the field?
Okay, so let's get granular. If FoxP3 is the master switch that turns a regular T cell into a peacekeeper, a Treg, what exactly is in their playbook? What makes them so powerful at suppression? Because it's clearly not just one single strategy. They seem to have this whole versatile arsenal.
They do. They use multiple dynamic, context-specific mechanisms. They don't just stand there and block things. They actively engage and disrupt the aggressive T effector cells. We call them TEFs, and the cells that activate them, the antigen-presenting cells, or APCs.
Let's break down their methods. Let's start with the one I find most clever. It's like an economic strategy, which you've called metabolic warfare, or the resource thief.
This goes right back to that uniform patch, CD25. Remember, we said Tregs express extremely high levels of CD25.
Which is part of the receptor for IL-2.
The IL-2 receptor, exactly. And interleukin-2, or IL-2, is the primary growth factor. It's the signaling molecule, the essential currency that those aggressive TEF cells need to survive, to proliferate, and to carry out their attack.
So when TEFs get activated to fight something, they start making their own IL-2.
They produce a little bit of IL-2, and then they need to consume it to grow their ranks and build an army.
But the Tregs are standing right there.
The Tregs are standing right there, and thanks to their hyperexpression of CD25, they have an infinitely higher affinity for IL-2. They're like highly efficient, aggressive resource gatherers in a video game economy.
Oh, that's a great analogy.
They just instantly bind and consume that precious IL-2 currency way faster than the TEFs can.
So they steal the food right off the TEF's plate.
Exactly. The Tregs don't even have to fight the rogue T cells directly. They just starve them. They deprive the TEFs of the survival signals they need, which causes them to undergo apoptosis, or programmed cell death.
So it's this really elegant negative feedback loop to make sure no T-cell response is allowed to spiral out of control.
It's brilliant. They win the war without firing a shot, just by controlling the supply chain.
Okay, so that's tool number one. What's the next one they pull out?
Next up is a strategy we can call metabolic conversion, which acts like the tranquilizer. This involves some pretty complex biochemistry using what are called ectoenzymes.
Whoa, whoa. Ectoenzymes. Let's slow that down. What is an ectoenzyme in plain English?
Good question. They're basically molecular factories that are located on the outside of the cell membrane. Tregs have two key ones, CD39 and CD73.
Okay, so factories on the outside of the cell, what do they make?
Well, it's more about what they convert. When aggressive TEF cells are fighting, they release a lot of a molecule called extracellular ATP.
ATP, I know that one. That's the energy currency inside the cell. So what happens when it's outside?
When it spills outside, it acts as a classic BAMP, a danger-associated molecular pattern. It's a loud biochemical alarm signal, screaming to all the surrounding cells, "Danger here! Increase inflammation! Everybody fight!"
So the fighting T cells are generating the very signal that tells everyone else to keep fighting. It's a feedback loop.
Right. And here come the Tregs with their little CD39 and CD73 factories. CD39 takes that pro-inflammatory ADP and breaks it down into an intermediate product. And then CD73 finishes the job. It converts that intermediate into an immunosuppressive molecule called adenosine.
So the Tregs are literally chemically cleaning up the alarm signal. They are transforming the very fuel of inflammation, ATP, into a molecule of peace, adenosine.
Precisely. This adenosine then binds to receptors on all the surrounding immune cells, both the TEFs and the antigen-presenting cells. And it creates this locally immunosuppressive microenvironment. It's like a targeted, localized chemical tranquilizer that just dampens all the activity in the immediate area.
That is just. It's incredibly sophisticated. So they manage the resource economy and the danger signaling, but they also have direct mechanisms, right? Physical interference, the checkpoints, and direct disruption.
Yes, they can physically interfere with the whole activation process. Tregs use key checkpoint molecules. You may have heard of these from cancer therapy, notably CTLA4 and LAG3, to interact directly with the antigen-presenting cells.
And the APCs, those are the generals. They're the ones that capture an antigen, like a piece of the pancreas they shouldn't be attacking, and then they present it to the soldier T cells, the TEFs, and say, go attack this target.
Correct. And for a TEF cell to be fully activated by an APC, it requires two signals. Signal 1 is seeing the antigen, but signal 2 is a crucial co-stimulatory signal, often from molecules on the APC surface called CD80 and CD86. Think of CD8086 as the official go button, the final launch key.
Okay, so what does the Treg do?
The Treg uses its own checkpoint receptor, CTLA4, which has a much, much higher affinity for CD8086 than the TEF cell's own receptor does. So the Treg basically swoops in, binds to the APC via CTLA4, and physically blocks that necessary co-stimulatory signal. It strips the APC of its go button.
It's like a security guard stepping in and jamming the radio just as the general is giving the command to attack their own teammates.
That's it. By blocking CD8086, the Treg forces the APC into what we call a tolerogenic phenotype. It essentially teaches the general not to activate the troops against that target.
Okay, and if all of that fails, the starvation, the tranquilizers, the radio jamming, if all those nuanced strategies fail, they can just resort to outright destruction.
They have the nuclear option, yes. Tregs can use nylidic enzymes, specifically granzyme B and perform, to directly induce apoptosis or cell suicide in targeted aggressive T cells and even B cells. If suppression fails, they just execute the rogue element.
What's so fascinating about this whole playbook is how versatile it is. You mentioned before these specialized gut guardians. I mean, the gut is a constant immunological war zone, yet somehow we maintain peace there every single day.
The gut is the ultimate test of peripheral tolerance, isn't it? It's constantly exposed to trillions of microbes, new food antigens. If tolerance breaks down there, you get severe inflammatory bowel disease.
So what makes the Tregs in the gut different? Are they a special class?
They exhibit incredible plasticity or adaptability. In the gut, you find a specialized subset called RO-ROT plus Tregs. And what's truly remarkable is that their existence and their function are strongly influenced by the environment, specifically by our diet and our microbes.
Wait, so their job security depends on what I had for lunch yesterday?
Essentially, yes. They are dependent on short-chain fatty acids, or SCFAs. SCFAs are the essential molecular byproducts that are generated when our gut microbiota ferment dietary fiber. They're basically the leftovers from the bacterial feast on the fiber you eat.
So let me get this straight. Eating fiber feeds the good bacteria in my gut. Those bacteria then generate SCFAs. And the SCFAs are the signal that programs these specialized Tregs to maintain peace in the gut.
That's the connection. These RO plus Tregs are crucial for establishing what's called oral tolerance. That's what prevents you from having food allergies and adverse reactions. And they specialize in suppressing these highly aggressive pro-inflammatory Th17 cells that cause a lot of tissue damage in the gut.
Wow, is there proof of that?
Oh yeah. Studies in mice show that if they lack these SCFA-dependent Tregs early in life, they develop devastating colitis the very moment they're introduced to solid foods. The diet microbiome axis directly programs our autoimmune security guards.
That brings us back to the master switch, FoxP3. The fact that the environment can dictate the specialized function of the Treg-like, whether it becomes an SCFA-dependent gut guardian, that suggests FoxP3 isn't just a simple on-off switch, is it?
That's the cutting-edge nuance of the field right now. The initial discovery viewed FoxP3 as the single, supreme boss of the cell's fate. But the new model suggests FoxP3 functions more as a transcriptional cofactor.
Okay, let's break that term down. Transcriptional cofactor. That sounds like some high-level biochemistry. What does it mean for the cell?
It means that FoxP3 doesn't always just bind to the DNA directly to turn on or off all the Treg-specific genes. Instead, it often requires the presence of other proteins, other DNA-binding partners, to stabilize it, to help it find the right spot on the DNA and to exert its control.
So FoxP3 is the manager, but it needs specific instructions or maybe specific keys from the environment to unlock its full potential in that location.
Exactly. Its interaction with the cell's internal machinery is heavily influenced by environmental cues. So when SCFAs from the gut microbiome are present, they activate certain pathways that create the necessary cofactors. Those cofactors then allow FoxP3 to turn on the ROROT plus genes and all the other gut-specific suppression pathways.
And without that input.
Without that cofactor input, the cell can still be a Treg, but it loses that specialized local function. It won't be a good gut guardian.
So this explains the versatility. The Treg isn't a static peacekeeper. It's an adaptable special forces unit. Its playbook changes based on the precise battlefield, the gut, the joints, the pancreas. And that is crucial for understanding how to actually fix autoimmunity.
This profound understanding that immune balance is maintained by these specialized, adaptable FoxP3 plus peacekeepers has completely redefined immunology. It's why we're finally moving past that terrible, scorched earth, non-specific immune suppression.
We've moved from trying to weaken the entire immune system to figuring out how to restore the underlying failure of tolerance using precise Treg-targeted therapies.
We have several strategies that are already moving through clinical trials, all of them exploiting the Treg's unique biology. Let's start with the most straightforward application. Low-dose IL-2 therapy.
Okay, so this targets the Treg's high expression of CD25, that IL-2 receptor we talked about.
Correct. Since Tregs are defined by their hyperaffinity for IL-2, they are exponentially more responsive to low levels of that cytokine than the inflammatory T cells are.
So the dose is everything here. If you give too much IL-2, you accidentally activate the entire rogue army again. But if you give just a tiny ultra-low dose...
You selectively expand the beneficial Treg population. You're essentially throwing a targeted, exclusive party just for the security guards, prompting them to proliferate and increase their numbers so they can better police the rogue cells.
And this is working in people.
Studies have shown really promising potential in diseases like SLE and type 1 diabetes. They've managed to restore a degree of tolerance without causing systemic inflammation. It's a fantastic, targeted way to use a natural growth signal.
Okay, so the next strategy is much more dramatic. It's not just boosting, it's cellular therapy, adoptive transfer.
This is essentially high-tech replacement, or augmentation. What we do is take a sample of the patient's own Tregs, expand them massively ex vivo, which just means growing billions of these cells in a highly specialized lab, and then we infuse them back into the patient.
That sounds incredibly complicated from a logistics standpoint. You're banking and growing live cells for a person.
It is very complex, but the trials have confirmed this strategy is safe and tolerable. In early human trials for type 1 diabetes and Crohn's disease, they've infused doses reaching, get this, 30 million cells per kilogram of body weight.
Wow.
And the early results are hugely encouraging. Some T1D patients are showing signs of preserved pancreatic function and even clinical remission after a year. It proves that simply boosting the numbers of the good guys can reset the entire balance.
But the drawback there, I would assume, is that those Tregs they're polyclonal. They're nonspecific. They're just general peacekeepers. So they might put down a riot in the joints, but they might also accidentally cause tolerance where we need a strong response, like to a new vaccine or an infection.
That is the major challenge of polyclonal transfer. It works, But it's still a bit like sending in a general security force for a hostage situation. That's why the next frontier is so exciting. Engineered Tregs.
So now we're taking a page right out of cancer immunotherapy. We're talking about CAR-Tregs and TCR-Tregs.
Exactly. Instead of infusing billions of general peacekeepers, we genetically modify the Tregs to target specific self-antigens, the exact protein targets that the rogue T cells are attacking.
So if I have type 1 diabetes, you would engineer my TREGs to only recognize the specific proteins on my pancreatic islet cells that the rogue Tefs are attacking.
Yes, we attach a specialized receptor to the Treg, either a chimeric antigen receptor, CAR, or a specific T-cell receptor, TCR, that directs the Treg only to the site of the attack. This is moving from a broad approach to what you call the heat-seeking missile.
So if they are that precise, what's the risk? I mean, what if they misfire or if they become too good at their job, too tolerant? If we program a TREG to specifically suppress T cells attacking a certain target, could we promote tolerance to a necessary foreign threat, like a bacteria that just happens to look similar?
That is the paramount question in the field right now, risk management. But the efficiency gain is it's mind-boggling. Antigen-specific Tregs dramatically outperform non-specific ones. For instance, in mouse models, it took only about 2,000 antigen-specific TCR Tregs to prevent type 1 diabetes.
What's in thousands?
2,000, compared to the millions and millions of non-specific cells you'd need. When you have surgical precision, you can minimize the dose, which inherently minimizes the risk of off-target effects.
That is incredible efficiency. It allows medicine to be highly targeted, halting disease progression at the cellular level with minimal systemic consequences.
And if we connect this back to the bigger picture, this is the profound philosophical shift that the Nobel Prize really acknowledges. We're treating chronic destructive diseases not by fighting the body, but by intelligently reminding it how to heal itself, or rather, how to regulate itself.
And the final fascinating application of this is finding a way to deliver this piece without even needing the whole cell. You're talking about exosomes.
Yet Tregs don't just work by touch. They can communicate long distance. They secrete these tiny lipid-bound vesicles called exosomes. These are essentially small, stable packages that carry the Treg's immunosuppressive cargo.
Like what?
Things like microRNAs or those CD73 ectoenzymes we discussed that make adenosine.
So they're like little cellular email packages containing instructions to calm down the inflammation.
Precisely. They mimic Treg function without needing to infuse the cell itself. And this is huge for clinical logistics. Exosomes are highly stable, they can cross tough biological barriers that cells can't, and they have an improved shelf life. Imagine a freeze-dried, cell-free preparation of peace instructions sitting on a pharmacy shelf, ready to be infused to restore tolerance.
So the 2025 Nobel Prize recognizes that true immune function is defined not by how hard it fights, but by how skillfully it maintains balance. We identified the Peacekeepers, we understood their instruction manual, which is FoxP3, and now we are literally programming them to treat everything from type 1 diabetes to lupus.
And that programming extends way beyond classic autoimmunity. Tregs are crucial in tissue repair and maintaining immune privilege during pregnancy, and they profoundly modulate inflammation in the nervous system.
So if we can effectively program these cellular security guards to restore order, the implications for neurodegenerative diseases that involve chronic inflammation, like ALS or maybe even certain models of Alzheimer's, are just huge. Imagine treating systemic, chronic, destructive diseases, not with blunt instruments, but with targeted, restorative balance.
And this raises a really important question for you, the listener, to hold out a mull over. We are currently figuring out how to precisely program our tolerance mechanisms to be laser-focused on these self-targets. If we succeed, if we create highly efficient, antigen-specific Tregs that can keep the peace everywhere, is there a hidden cost? Could maximizing our capacity for tolerance against self-attack inadvertently limit our necessary immune surveillance? Could it allow a very subtle, unrecognized cancer or a novel pathogen to gain a foothold, simply because our cellular security guards have become too good at maintaining the peace?